Okay, so we're going to talk about the microbial disease project that you'll be doing this semester, and that is in modules. And this is, I get on student view here, so we shouldn't have to scroll very far to find the project. Okay, then click on the disease project link here, and that'll get us to several things. So you've got the project instructions, which I'll open up here a second and we'll go through. And there's a extensive list of microbial diseases to choose from on that link. And then there's a checkoff list. As you get going along there's a checkoff list. I'll show that here in a little bit, for you to check and make sure you're hitting all the requirements. And then there's the grade rubric for you to look at as grade the presentation and talk. And then there's the sign-up sheet for your disease and disease-causing organism. Okay, so let's take a look at the project handout. And so I'm going to make that here a little larger, which it loads up. I'm a little slow today. Okay, so what you're going to be doing this semester is you're going to pick a microbial disease, and it could be bacteria, it could be fungi, it could be a protozoan, could be a parasite like a roundworm parasite like a nematode, or hookworm, dog heartworm, and it can be, for those of you who have any vet tech people, any vet tech, okay. A lot of times the vet tech people like to do animal diseases, and that's fine. Or a flatworm like a tapeworm or fluke. So any relatively microbial disease will work. Okay, let me get that out of there. You're going to research it, and there's a couple things that you need to research, and we'll talk about, or two parts to the research you're going to be doing. We'll get to that here in just a second. And then you're going to put that information that you're gathering on your research into a presentation, either Google Slides, PowerPoint, Prezi, whatever your presentation tool you like to use. And then you're going to write up your talk, what you're going to say for your presentation, and then you're going to present that. Now again, I'm doing this recording for both you guys face-to-face and online. So you guys will be face-to-face, you guys will be presenting during lab, and then for you online people that are going to be listening to this, you will schedule a time to present. And we'll talk more about that for online people down the road. All right, so the first thing obviously is to choose what disease and what organism is causing that disease. And so what I'd like to do is get that decided by Friday, September 9th. Again, there's a sign-up sheet for you to do that back under the projects link there. And again, it's going to ask you to put your name and the name of the disease and the name of the organism causing the disease. So give me if it's a bacteria, give me the scientific name of the bacteria. Or if it's a protozoan or a virus, give me the full name of that organism. And then now there may be some cases where the more than one organism may be involved in causing that disease. So in that case, just pick, pick the one that is most prevalent or most commonly associated with that. Again, you may mention that there's multiple organisms that are involved in that disease, but then you're going to pick one, because a part of the presentation involves you talking about the microorganism that's causing the disease. And so you'll need to just pick one that you'll do on that part. Okay, so once you selected the disease you're going to do and recorded the disease and the disease organism, then of course you can start doing your research. And there are two parts to this presentation. There'll be research that you're going to do on the organism that's causing the disease, and we're going to talk about, you're going to do some taxonomy, talk about the domain, kingdom, phylum, class, order, family, genus, species that it's taxonomically grouped into. And then you'll also talk about the group characteristics. And again, I'll have some examples of that here in a second. And then you'll talk about the morphology of that organism. It's the size. If it's bacteria, you're going to talk about several things. What is it? What's the characteristic shape of the bacteria? Does it have any cell arrangements like in pairs or chains or diplo or tetrads, groups of 4, and so on. And then other biochemical characteristics. Again, we'll look at. I have some examples down in the handout here in just a second. We'll look at. So that's the thing you'll talk about again about the microorganism causing the disease, and then you'll get into talking about the disease itself. So you'll have the introduction to, well, there could be the introduction. It depends. Some people do this. They have their introduction before they get into the organism that causes the disease, where they just give a couple sentence introduction to the project or disease. Some will do it after they talk about the microorganism, but you'll have a little introduction. Talk about signs and symptoms, pathogenesis, epidemiology, disease diagnosis, treatments and preventions control. Again, we'll get a little more into that here in just a second. And then you'll put together your presentation, and then you'll have a presentation talk. And the talk will be basically written out what you're going to say as you go through slide to slide to slide. Here's what I'm going to say for my first slide. Here's what I'm going to say on the second slide, and so on. Remember that the presentation is just a visual aid. So you're not going to have like paragraphs of information plastered onto the presentation. You'll just have kind of tidbits or highlights or things like that, and then you'll fill in the information with your talk. Does that make sense to everyone? Okay. And so that needs to be done. The presentation and the script needs to be done and uploaded on Canvas by Sunday, November 14th, midnight. And then we'll start in face-to-face. We'll start Wednesday, that Wednesday, November 17th, giving presentations or somewhere. We actually have a couple weeks or 2 weeks, 3 weeks reserved, I think, for presentations. So with you guys, it just kind of depends on, we could do it that Wednesday, we could do it the next week. So we'll talk about that more when it gets closer. And then, like I say, the online people, you guys will be signing up to present that starting that week of November 15th. Okay, so so here's some examples then what I'm looking for when you start talking about the specific areas. So first we're going to talk about the disease causing organism. So again, one of the things you have to do is the taxonomy. I'll jump down here to step 2. Excuse me. So with the taxonomy, you're going to look up and find what its classification groups it belongs to. So for everything except viruses, you'll have a domain it belongs in, kingdom, phylum, class, order, family, genus, species, and then its scientific name. Okay. So for example, let's say that I'm doing gonorrhea. And if I was doing gonorrhea, then the organism that causes gonorrhea is in this area, gonorrhea. And so here is these taxonomic groups. It's in the domain bacteria, kingdom eubacteria, phylum proteobacteria, so on and so on and so forth. Now that seems like Mr. Hagee, Professor Hagee, that sounds like that's going to be hard to do, to find that crap. Well, it's really not that hard. Believe it or not, Wikipedia does a pretty good job on taxonomy for the most part. So let's just show you that here. So I'm going to take, so if you go and search this scientific name of your disease organism, or the virus name if it's a virus, one of the things you're going to find is the Wikipedia page. And so if you go to the Wikipedia page, it's got some of it there, but let's go right here. Click on the Wikipedia page, and notice on the right-hand side there, or yeah, right-hand side, we've got its taxonomy. Now notice though that this one left out the kingdom. Okay, so if you do any bacteria, the bacteria are in the domain bacteria, unless you do an archaebacteria, its own domain bacteria, and then the kingdom is eubacteria, but they didn't list that. Okay, so it's not perfect, but it's pretty good, you know. Now sometimes you'll find on a bacteria search they'll say domain bacteria, kingdom bacteria, but technically it's eubacteria. You know? And so there's the phylum, class, order, family, genus, species, and specific epithet, Neisseria gonorrhoeae. Okay. Now the other part that I want you to do, before we do that. So if it's viruses, you can search the same way, and you're probably going to find a mixture of the Baltimore and the ICTV classification. But you should be able to find, you know, it should show the group, order, family, subfamily, genus, and then the specific name. And so I did chickenpox, and chickenpox is caused by the herpes virus and the HHV3. And so it's group 1, it's in the order Herpetviridales, it's family Herpeviridae, alpha Herpeviridae, and so on. Okay. Then, like I say, if it's anything else other than a virus, then you'll have a domain, kingdom, phylum, class, order, family, genus, species. So, for example, here's malaria. The Plasmodium vivax is one of the strains that causes malaria, and there's its group. Okay. Then the other thing about the taxonomic groups is I want you to tell me, so what does it mean to be in the domain bacteria? What does it mean to be in the kingdom eubacteria, and so on? Now, if you go back to your Wikipedia page, that will also help you pretty much. So if you click on domain, it's going to give you, well, right there, it gives you the characteristics of what it means to be in the domain bacteria. Proteobacteria gives you the characteristics there, and so on and so forth. And you could also click on that and it would go to that specific page. So anyway, use Wikipedia to help you on taxonomy, okay? And for those two parts. Then for morphology, the last thing with the disease-causing organism would be the morphology. Okay, and here's an example of the taxonomic characteristics for each of the taxonomic groups for gonorrhea there. Okay, so for bacteria, you know, tell me what's the size, you know, what is it, gram positive, gram negative, what this type of cell shape, is it rod shaped, coccyx, spirilla, so on. Do any cell arrangements like pairs, diplos, tetrads, clusters like staphylo, or chains like strepto. Colony morphology. We're going to do a little bit with that next week, or no, the week after. But we'll do some next week. Colony morphology would be, what's it look like when you grow the bacteria on an agar plate? What's the colony characteristics? And there's specific terms for the overall shape and the edge and the elevation and so on and so forth. So if you're doing bacteria, you'll give me some characteristics there. To produce endospores, is it acid positive, negative? Is it does that have a capsule, and so on. And then there's some examples for virus, morphology of the virus. And then you'll do some similar things for if it's a fungi or protozoan or a eukaryote. Tell me about the morphology. Okay, so that's the first part, or part 1 there, the information you're gathering to put in your presentation. And then part 2 is over the disease. So again, an introduction little introduction. And then again, some people do the introduction, or most, I guess, usually they do the introduction before they get into the disease-causing organism characteristics. But just a little bit, what name of the organism, name of the disease, name of the organism, maybe some interesting facts, maybe what body system does it infect, and so on. So just a little introduction, paragraph. Signs and symptoms, so listing out and discussing any of the signs and symptoms. So the pathogenesis would be probably one of your bigger, or if not the biggest, section. We're going to talk about how do you get that disease? What's the reservoir? What's the vector of transmission? How do you pick it up? How do you get it? How does it enter the host? So what portal of entry? What does it come in through the respiratory system and break in the skin, through the urogenital tract, or so on? How does it get into the body? What's its portal of entry? What does it do once it enters the host? That's going to be the big part. How is it causing damage, or how is it causing the signs and symptoms? Is it producing a toxin? Is it disrupting cell function? Is it stopping mucus production? What is it doing? What's the pathogen doing to cause the signs and symptoms? How's it damaging the host? I kind of have that listed there a couple times. You may find some things about ways that the pathogen avoids the host immune response, the non-specific immune response. Maybe it hides in blood clots, or produces some enzymes that prevent enzymes from attacking it from the host, and so on. And that also gets into, what is it doing to avoid the non-specific response? What's it doing to avoid the antibody production? What's it doing to avoid cell-mediated response? Not all pathogens have methods to avoid those things, but kind of look to see if yours does. Okay. A little bit of the life cycle. Definitely if it's a virus, or if it's a vector-borne pathogen, so like if you get it from a mosquito bite, tick bite, whatever, you should have kind of a life cycle showing the infectious cycle there. You can even have that with bacteria. It's showing the infectious cycle with how it's contacted and maybe how it's spread. So kind of look for those things. But you'll definitely have that with virus. You'll definitely have that with any parasitic diseases, any vector-borne diseases. Should have a life cycle. Okay, epidemiology. So on the epidemiology, there's a lot of things that could be in there, but what I want you to kind of focus on is the occurrence. Where is it most common? What's the rate of occurrence? Like some statistics, like how often is it showing up in populations? Could be in the U.S., could be worldwide, could be both. Are there any groups that are more susceptible to it? What are the risk factors? So those kind of things with epidemiology. Diagnostic tests. So what do they do? What tests do they do to determine that you have that illness? And what are the tests looking for? Are they looking for certain proteins? Are they looking for toxins? So what tests are used, and how the tests work, or what are the tests looking for? Treatments, cures. So what are the chemotherapeutic agents? Are they using antibiotics? Giving monoclonal antibodies, what do they do to treat it? If there are drugs being used, what do the drugs do to kill the pathogen or harm the pathogen? Especially if it's antibiotics. What's the antibiotics' mode of action? How is it prevented and controlled? There are vaccines, and so tell us about the vaccines. What are the things that they do to try to prevent the spread and control the disease? Economic impact. So what impact does having this disease cause? And this could be, it could be something to do with the fact that it's like worldwide, or whatever it is, that they're spending a lot of money to cure it, treat it. The medication is costly. Or it could be where you might just have to speculate, okay, all right, if I get this disease and end up in the hospital, average hospital stay is X amount of dollars, treatments are X amount of dollars. So, so you may have to do some speculating as far as the impact of having that disease. And some places you'll find statistics that'll tell you that, like the annual cost of this disease is X millions of dollars, whatever. And then your last slide will just be a little conclusion, a little summary slide to kind of tie things together and end your presentation. Okay, so far so good. You know, we're kind of running through that. Seems like I'm flying through that. Okay, so this citing of sources. You need to definitely have a works cited on that you'll either put as a slide, the last slide, I guess, after the conclusions in your presentation, or you could put it in your talk that you're going to submit at the end of your talk where you're going to submit it. But any sources you use to gather your information, please cite. And then just use bibme or easybib.com or whatever you use to put your sources in the proper format. Okay. You don't have to do any in-text citations or anything like that.
This doesn't, they're not APA.
Not other than, I mean, you can use APA to make your works cited page, but you're not going to put any. The only citation you're going to put in your presentation is any pictures, figures, diagrams that you get off the internet. Then just put a text box underneath that diagram and put the website link. Otherwise, you'll just have a works cited page. Okay. And then I talk about that for the presentation. Yeah, so then gather your information and start preparing your presentation. And again, remember that the presentation is your visual aid, the body of information you're going to be giving during your talk, the oral talk. And as it says there, your visual aid presentation should not contain complete oodles of information, big paragraphs of information. It just should be bullets, and you're going to have pictures and diagrams and those types of things to make your presentation presentable. Nice, make a nice presentation with some visual aids there. Then again, you'll have your script, writing your script, which is again what you're going to say for each slide as you go through the slideshow. And you'll have that script with you, if however you want to have that script as a handout or on note cards or whatever when you give your talk. So you have that with you. Again, you want to rehearse it and make sure it flows smoothly and stuff. And a lot of times memorize it, but you always should have your script with you there in case you get off track. Okay, like I say, presentation is pictures and figures as a visual aid. And then you'll cite those within the presentation, just using a text box to cite any diagrams, graphs, life cycles, so on. Okay, and again, just reminding you, did I misspell Wednesday? Oh, just a paragraph or a comma. Upload the presentation and your talk by 17 or the 14th, Sunday, November 14th. And then class will start probably on that Wednesday. Okay, a little bit about the format. Again, on your presentation, again, make sure that the things that you do have, the text that you have is large enough and easy for people to see. Make sure that colors contrast so you can see the text and stuff nicely. Yeah, and that's just talking about your presentation talk as far as being at least 12 point font. Easy for me to read. And you can make it as big as you want, I guess, if it's easy for you to read. So this is just kind of showing the outline format here. So again, giving you some little tidbits of what it should be. So you'll have your introduction, and you should title your slides. Like if this is your introduction slide, of course you'll have your title slide. Then you'll have if your next slide is your introduction slide, then you should have title introduction, and then your introduction. Title disease causative agent, and then things that you're going to cover there. signs, title, signs and symptoms, title, pathogenesis, and so on. And there's a few links to maybe help you get going. If we go back to the project on modules here, the checkoff list. So when you're kind of getting done or going along here, you open up that checkoff list, and then that's pretty extensive there. Those are the things that you should be having in your presentation. So just you can refer back to that checkoff list for your FYI to help you there. And then the rubric. Again, remember that if you, for some reason, choose not to present, then that's a 40-point deduction. So you need to present. Okay. I know not everybody likes to present, but we're going to try to make it as painless as possible for you. So don't worry. There's just the rubric that I'll use to score. All right. Any questions at this point in time? Okay. Now remember that I'm here to help. So if you're going along and you're not finding something, you're having problems finding something, then get a hold of me and let me know, and I'll see what I can help you. I'll help you out. That's what I'm here for. So don't don't worry if you're having problems finding something. Let me know. I'll help you out. All right, so I'm gonna stop screen share and end this recording.